I'm Amanda Weiss, and today I'm going to talk a bit about clonal selection of B cells in the animal immune system. The purpose of the immune system is to protect the body. If a pathogen manages to get past the first line of defense, the body can then produce an adaptive response, which involves B cells and T cells, in order to stop the pathogen from causing more harm. One of the primary aspects of adaptive immunity is that it is specific. The immune system can react to specific pathogens that enter the body. Pathogens can be identified by their antigens, which are specific non-self substances often attached to the cell membranes of the pathogen. The body is capable of producing many different antibodies which can match the specificity of these antigens. As part of the immune system, an animal's body produces many different types of naive B cells, which each display the specific antibody that they can produce on their cell surface. When a pathogen enters the body, an act of recognition occurs. The antigen binds to its specifically matched antibody on one of the specific B cells. A T cell then comes along and activates the B cell, which bound the antigen and thus the pathogen. This stimulates the B cell to produce a clone of identical cells, all of which are identical to that original B cell. Some of the cells in the clone go on to become plasma cells, otherwise known as effector B cells, which then produce and secrete the type of antibody which bound the antigen in the first place. This is the effector stage of the immune response, as it brings into effect the defensive antibody production. The antibodies can then go and bind the pathogen elsewhere in the body by the antigens on their surface. The clone cells which did not become plasma cells develop into memory cells, which will then be able to respond more quickly in the event of the pathogen invading the body again. This more rapid response would be referred to as a secondary immune response. The memory cells do not secrete the antibody, and they divide slowly in order to persist for a long period of time so that the body has immunity from the pathogen for a long period of time. This entire process of clonal selection helps defend the body against highly specific pathogen invaders.